Good morning, students. Welcome back again to another session of plant physiology. Chapter number 4, photosynthesis in higher plants. So far, you have learnt about the site of photosynthesis, PS1, PS2, photosystem 1, which is located at 700 nanometer wavelength, photosystem 2, which is located at 680 nanometer wavelength, and the light reaction that is taking place in the granum thylakoid, and how the light is getting harvested, and how the light energy is shifted to ATP and NADPH. ATP, NADPH, and oxygen are the end products, and these end products are very important. As you know, oxygen, it is very important for respiration, which is a gas getting diffused out of the chloroplast. And ATP and NADPH, these are synthesized in the stroma. The light is harvested in the grana, but the synthesis takes place in the stroma of the chloroplast, where further these ATP and NADPH are going to get utilized for carbon fixation. Carbon dioxide and water will be utilized, and carbon dioxide is going to get synthesized into a glucose molecule. And this glucose molecule is very important for life to exist on Earth. And that's why the carbon fixation phase is also called as biosynthetic phase. The light reaction, the photochemical phase, and the carbon fixation, the biosynthetic phase. So today, the very important topic, the Calvin cycle, is going to be discussed in this session. I hope you will be following clearly everything without any confusion. Let us hope so. And before that, I'll go to my favorite part of this session, that is appreciation. I would like to appreciate Abhinaya Sri G. Archana from Vangara TSRJC, Y. Nagasri S. Akhila R. Triveni from Vaira TSRJC, M. Sadguna B. Ashpita from Tatipelli TSRJC. Very good, students. You are doing very well. You are finishing the assignment in time. In the afternoon session, you are finishing and forwarding it as early as possible to your JLs, and they are also doing the same work. They are passing it to me, and I feel very happy because whatever I am teaching, it is going into your brain, and you are able to write the assignments in time. Those assignments which are very important as per IPE point of view. I hope all other students are also following the same thing and doing the work in time and keeping with them. Whenever your jails ask, provide it to them. So now move on to the topic. Where are the ATP and NADPH used? As you know, in the light reaction, you have seen ATP synthesis and NADPH is done. And this is done in the stroma of the chloroplast. Where are they used? They are used in the biosynthetic phase. The biosynthetic phase where carbon dioxide will be fixed, getting converted to sugar. So here ATP and NADPH are used to drive the processes leading to the synthesis of food, more accurately sugars. And of course, this is a biosynthetic phase, which is very essential for life to exist on planet. This process does not directly depend on the process light, on the process of light. So here we generally say it is a Dock reaction. I think it is a misnomer to say Dock reaction because this process does not directly depend on the process of light, but is dependent on the products of light reaction, that is the ATP and NADPH, besides carbon dioxide and water getting utilized here. So immediately after light becomes unavailable, when light reaction will take place during the light availability, and when the light becomes unavailable, the biosynthetic process continues for some time and then stops. Why it will stop? Because when light is there, then only ATP and NADPH will be synthesized. When this is there, then only the biosynthetic phase will be continuing. So as on the ATP and NADPH will be utilized, the biosynthetic phase will stop. And then light is made available, then the synthesis starts again. So it is better to call it as a biosynthetic process, biosynthetic phase, than to say simply a Dock reaction, as we usually became uh, habituated to call it as a dark reaction. So coming to the primary acceptor of carbon dioxide, as in this reaction that takes place in the stroma of the chloroplast, carbon dioxide is the raw material getting utilized now in this biosynthetic phase. Who is going to accept this carbon dioxide? That will identify. So Melvin Calvin, scientist, explained dark reaction using carbon 14 pathway. Radioactive isotope, he utilized carbon 14, and in that pathway he identified who is going to accept this carbon dioxide molecule. What he did, he killed green alga using hot methanol. Green alga, it will be undertaking photosynthesis, and when it is killed, what will happen? It is easier to find out whatever the products present in the cell. So he examined the products in the cell after killing with methanol. In the cell, he found certain substances. He found the first compound was a carbon compound. So some stable carbon compound he found, that is phosphoglyceric acid. This phosphoglyceric acid, in short, we'll say PGA, it is having 3 carbon and carbon dioxide having 1 carbon. That means the 1 carbon is going to combine with a carbon which is having a compound which is having 2 carbon, then only it is possible to give a 3 carbon compound. And as Melvin Calvin identified phosphoglyceric acid, it is a 3 carbon compound. So they were thinking there may be some 2 carbon compound existing that is combining with this 1 carbon of carbon dioxide gas and forming the PGA, phosphoglyceric acid. So they thought so, but it was not so. It was wrong. As you can see, carbon dioxide plus 2 carbon compound will give 3 carbon compound, maybe that is PGA. And some 2 carbon compound we have to identify, they thought. And when they identified, they did not find any 2 carbon compound. Instead, they found a 5 carbon compound. This 5 carbon compound is combining with this carbon dioxide, then only the phosphoglyceric acid is going to form. So here we can see 1 carbon of carbon dioxide combining with a 5 carbon compound. 1 plus 5, it will be 6. 6 carbon compound. This 6 carbon compound hexose sugar is unstable and it is splitting into 2 molecules, each one having 3 carbons. And this 3 carbon is only our phosphoglyceric acid. And that's why it is written here 2 PGA. That means 2 molecules of phosphoglyceric acid is going to form whenever 1 carbon compound is combining with a 5 carbon compound. So this was clear. So I think for you also it is clear that the acceptor of this carbon dioxide 1 carbon is none other than a 5 carbon compound, and then it is unstable, it is splitting into 2 molecules of phosphoglyceric acid. So in this way we'll proceed here, and you can see it was identified RUBP. This RUBP is nothing but the 5 carbon compound. This 5 carbon compound is none other than RUBP, ribulose bisphosphate. Bisphosphate means biphosphate. 2 phosphates will be there. 2 phosphates will be there. You have to remember this thing about RUBP. So here RUBP is having 5 carbon. It is a ketose sugar, and it is also having bisphosphate means 2 phosphates. So RUBP is a ketose sugar. I hope up to here it is clear for you that our carbon dioxide is going to get combined with a 5 carbon compound RUBP. So we must know in detail about this RUBP, how it is combining with carbon dioxide. Some enzyme is present, that enzyme we are going to discuss. And the process is called carboxylation. Carboxylation because carbon dioxide combining with a acceptor molecule, 5 carbon acceptor molecule RUBP. So this process is nothing but carboxylation. The term is new for you. So here carbon dioxide and RUBP, 1 carbon and 5 carbon is going to give 3 carbon compound. How many molecules? 2 molecules. 2 molecules of phosphoglyceric acid is going to form. So here the most important thing is Rubisco. Rubisco is an enzyme, ribulose carboxylase oxygenase. Why the name is given means Rubis. Rubis means it is working on RUBP. Okay, ribulose, it is working on RUBP. So Rubis, and this C and O is nothing but carboxylase oxygenase. Carboxylase oxygenase. I am mentioning here carboxylation because RUBP is combining with carbon dioxide. But oxygenase nature is also there for this Rubisco enzyme. Remember this important point about Rubisco, that Rubisco is 2 in 1. It is going to have a carboxylase nature and oxygenase nature. Carboxylation whenever carbon dioxide is available, oxygenase nature whenever oxygen is available. That's why the name of this enzyme is Rubisco, and it is the most abundant enzyme on earth. You can see I have given here this image of Rubisco. The nature of Rubisco is carboxylase oxygenase. Carboxylase that can be seen when it is combining with carbon dioxide in the photosynthesis stage 2. That means biosynthetic phase. And it is for energy production. The other part of this Rubisco, it is also having oxygenase. The oxygen molecule is going to get combined with RUBP, and it is going to undertake photorespiration in the presence of sunlight. Respiration is also carried in the plant cell, that is called photorespiration. And here the waste product, because it is of no use for this plant, so it is considered as waste product here. So the nature of Rubisco can be understood by this image clearly. Rubisco is carboxylase as well as oxygenase. Carboxylase for photosynthesis, oxygenase nature can be seen in the photorespiration. In coming topics, you will learn about this nature also of Rubisco enzyme. Now we have to focus on this only. Rubisco enzyme is helping RUBP to combine with carbon dioxide and form phosphoglyceric acid, and the entire process is called carboxylation. So the same thing is given here in this image also, how the ribulose bisphosphate, RUBP, as I said, it is having 5 carbons, and bisphosphate means 2 phosphates. It is combining with carbon dioxide, and who is helping here? The enzyme Rubisco enzyme is helping, catalyzing it, and here 2 molecules are formed. Which 2 molecules are formed? 3-phosphoglycerate, or simply I said PGA, phosphoglyceric acid. Don't get confused with the names here. Phosphoglycerate also same only, phosphoglyceric acid also the same only. And why we are mentioning here 3 means it is having 3 carbons, and you can see 3 phosphoglycerate is written and 3 phosphoglycerate is written. That means 2 molecules of phosphoglycerate are formed here, and it is having 3 carbon 3 carbon atoms, and 1 p is also mentioned here. 1 p means, as you know, Rubisco is having 2 phosphate here. It will also divide, no? So when it is dividing, 1 phosphate for this PGA, another phosphate for another PGA. So here 3C, 1P indicates 3 carbon atoms and 1 phosphate atom present in one single phosphoglyceric acid. So here 2 molecules of phosphoglyceric acid is formed. So this is carboxylation, the very important step that you have to remember. Photochemical equation. Photochemical equation, why I have given means just to have an idea in your brain. Through the entire class or session, you have to remember this. 6 molecules of carbon dioxide. Carbon dioxide is going to combine with 12 molecules of water in the presence of sunlight, means harvesting the sunlight. The chlorophyll molecule is synthesizing sugar, that is glucose, C6H12O6. 6 molecules of water and 6 molecules of oxygen gas is liberated. So this is the photochemical equation that we have already learned in 10th standard. But here, remember, how many molecules of carbon is going to participate? 6 carbon. From 6 carbon dioxide molecules, 6 carbon. And here also you have to remember how this is going to proceed in the biosynthetic phase, the carbon fixation, or we can also say it as the Calvin cycle, or the C3 cycle or C3 pathway. Why C3 pathway means 3 carbons. As I said, phosphoglyceric acid. So here already you are familiar with the. So here Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. Remember, in a cyclic manner, it is taking place round and round cyclic manner. That's why it is called as Calvin cycle, introduced by Melvin Calvin, for which he got Nobel Prize also. So here, why it is occurring in a cyclic manner for the RUBP generation? Again and again, RUBP must be made available for combining with carbon dioxide. So all the carbon dioxide molecules which are entering in the leaf, they must combine with RUBP. So RUBP must be available. RUBP must be available. So for this reason, in a cyclic manner, the entire Calvin cycle is going to be in a cyclic manner. And we can describe this Calvin cycle in 3 stages. One is carboxylation, reduction, regeneration. It is very simple and easy to remember all these stages. Carboxylation, as just now I have discussed, how carbon dioxide combining with RUBP and phosphoglyceric acid is going to form. Reduction. In this reduction, what is happening? Already you know, in light reaction, ATP and NADPH are synthesized. That will be utilized in the reduction. In reduction phase, the phosphoglyceric acid will be converted to aldehyde group, phosphoglyceraldehyde, or we can say glyceraldehyde 3 phosphate also. So in this reduction, ATP and NADPH will be utilized. And of course, the regeneration. Regeneration means again and again RUBP must be made available. That's why the phosphoglyceraldehyde which are formed, some of them is going to regenerate the RUBP back again. So this is very simple cycle involving 3 important stages. First stage, carbon dioxide combining with RUBP. Second stage, utilization of ATP and NADPH for making that PGA getting converted to aldehyde. And the third stage is the regeneration of RUBP. So this cycle is already given in your textbook, but here some change is there. Some change is nothing but here I am mentioning 3 molecules of carbon dioxide. For easy understanding, instead of mentioning only 1 carbon dioxide or 6 carbon dioxide, we can take 3 carbon dioxide molecules here. And you can see the carboxylation is the first stage. It is called carbon fixation. Carbon is getting fixed with some RUBP. Okay, and the second stage, as I said now, reduction, where ATP and NADPH will be utilized, which are the end products of light reaction. And meanwhile, what is happening from the second stage? It is moving here, the phosphoglyceraldehyde which is forming. Some of them are forming the sugar, and the remaining RUBP generation of RUBP. Why? Combine with another carbon dioxide molecule, and this cycle is going to okay in a cyclic manner. So here remember one most important thing, writing here 3 molecules of carbon dioxide, but in nature for every carbon dioxide molecule there will be a one Calvin cycle taking place. So 6 carbon dioxide are going to participate in the photosynthesis process for making one glucose molecule form, means 6 Calvin cycles will occur. This is the most important thing you have to remember. For every glucose molecule to form, 6 Calvin cycles will be there. every carbon dioxide molecule will be undertaking this Calvin cycle when it is combining with RUBP, when carboxylation will begin. Okay, so here, as you can see, Rubisco is the enzyme helping, and here, let me write here RUBP, ribulose bisphosphate, which is a 5-carbon compound, is combining, and here it is going to form 6-carbon compound hexose sugar, which is going to split into 2 molecules of phosphoglyceric acid. Okay, 2 molecules of phosphoglyceric acid is going to form. But 3 molecules of carbon dioxide I have mentioned here. So I must write here 3 molecules of RUBP also. So we'll see 3 molecules of carbon dioxide and 3 molecules of RUBP. Can you guess how many carbons will be there in 3 molecules of RUBP? If each RUBP is having 5 carbon, F, it is 3 5 ja 15. 15 carbons will be present. For 3 molecules of RUBP, 15 carbons will be present. And here 3 carbons of this carbon dioxide molecule. 15 plus 3, how many? Total 18 carbons. Total 18 carbons. And you know, it is going to split into phosphoglyceric acid. Phosphoglyceric acid, which is having only 3 carbon. That means we have to divide it with 3. That means 6 3 ja 18, no? That means 6 molecules of phosphoglyceric acid is going to form here. So at this point, 6 molecules of phosphoglyceric acid will form. 6 molecules of phosphoglyceric acid will form. Remember, 3 carbon dioxide molecules only we are taking. Means another 3 molecules of carbon dioxide also 6 PGA will form. 6 plus 6, 12 will be there. So for this cycle, we will remember 6 PGA having 18 carbons. On total, 18 carbons will be there. Now the next stage is reduction. In this reduction stage, what will happen? ATP will be added here. Why ATP will be added means, because this phosphoglyceric acid, it is having only one phosphate. Now it will become diphosphate. It will become 1,3-diphosphate. It previously only one phosphate was there. Now to the third ring of carbon, the another phosphate is going to get attached. So it is called now diphosphate. Having 3 carbons only. Having 3 carbon only. So it is called 3-glyceraldehyde phosphate. So ATP will be added next. That means it is going to become diphosphate. Then afterwards, it will become 1,3-diphosphoglycerate. Reduction will be taking place by addition of this NADPH. NADPH, which is also product of light reaction, will be added to reduce it, and it is going to give its hydrogen protons to this, and it is going to get reduced here, and it is going to get converted from acid to aldehyde. Acid to aldehyde. Now the name will be changing, and it is called as glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Or in some books it is given as PGAL. PGAL also, don't get confused, it is phosphoglyceraldehyde. PGAL in short they will be writing, or else glyceraldehyde 3-phosphate. I think now it's clear. After carbon fixation, addition of ATP and NADPH will convert the phosphoglyceric acid to phosphoglyceraldehyde, or we can say glyceraldehyde 3-phosphate. Now how many glyceraldehyde 3-phosphate are there here? 6. 6 glyceraldehyde 3-phosphate are there. These 6 glyceraldehyde phosphate, from this one is going to separate, and it is going to combine with another glyceraldehyde 3-phosphate. That means 2 glyceraldehyde 3 phosphates will be combining to form sugar here. Okay, so one is going to get separated. Remaining 5 glyceraldehyde phosphate will be there. Remaining 5 glyceraldehyde phosphates, they are going to lose 2 phosphates and they'll get converted to ribulose 5 phosphate. Ribulose 5 phosphate because having 5 carbon. Let us recall here the 18 carbons were there. From 18 carbons, 3 carbons are removed. That means 1 phosphoglyceraldehyde is removed. Remaining 15 carbons will be there. From these 15 carbons, they are having only single phosphate. From these 15 carbons, and they are having 5 phosphates. 2 phosphates will be lost. Here it is not mentioned. 2 phosphate will be lost, and it is going to become ribulose 5 phosphate. That means ribulose 5 phosphate having 15 carbons means 3 molecules will become here. 3 molecules of ribulose 5 phosphate will be there. For this single monophosphate, 3 ATPs will be added again to make it bisphosphate. And that bisphosphate is nothing but RUBP, ribulose bisphosphate, having 5 carbon each. So 3 molecules of such RUBP are going to form again here for this 3 molecules of carbon dioxide. So here 3 is going to form means in another 3 molecules of carbon dioxide, more 3. Totally 6 are going to form. So why we are taking here 3 molecules? If we take one single carbon dioxide, it will be very difficult to say about the phosphoglyceraldehyde. We can't say one PGA is going to. We have to write in fractions. So instead of going for fractions and rather taking all 6 carbon dioxide molecules, we'll take here 3 carbon dioxide, 3 RUBP, carboxylation. Okay, and after carboxylation, 2 molecules, we are getting here 6 molecules of phosphoglyceric acid. 6 molecules of phosphoglyceric acid by addition of ATP and NADPH is getting reduced. And after getting reduced, the acid is going to become aldehyde, that is glyceraldehyde 3 phosphate. And this glyceraldehyde 3 phosphate, 6 molecules from that one is given out for sugar formation, and the remaining 5 is going for the regeneration of RUPP. Here also, directly it is not going to become RUBP, it has to lose 2 phosphate. 2 phosphate it will lose. When it will lose 2 phosphate, only 3 phosphates will be there. 3 phosphates are there and 15 carbons, and that compound is now called as ribulose 5 phosphate or monophosphate. It is monophosphate. It want to become bisphosphate. So it will be getting added by ATP again. When ATP is added, the phosphate of the ATP will be taken and ADP is going to form again. So here 3 ATPs phosphate is getting added to this ribulose 5 phosphate, and it is becoming ribulose bisphosphate 3 molecules having 5 carbon each. So this is a cycle taking place, and for every carbon dioxide molecule, one single Calvin cycle is going to take place. So that's what we are going to have a clear understanding stepwise manner in carboxylation. The carbon atom of carbon dioxide is fixed by RUBP. A 6 carbon unstable compound is going to form. It is spontaneously hydrolyzed into 2 molecules of phosphoglyceric acid. So this is the thing, carboxylation. And here for your easiness, I am mentioning one single RUBP combining with single carbon dioxide, then 2 PGS will form. When 3 RUBPs are combining with 3 carbon dioxide, that means 6 PGA is going to form. In the previous slide, you have seen about this one only. 3 RUBP, 3 carbon dioxide, and 6 phosphoglyceric acid formation you have seen. But actually 6 RUBP, 6 carbon dioxide, and 12 phosphoglyceric acids are going to form. 12 phosphoglyceric acid are going to form. And remember one thing here, 6 PGA. From this 6 PGA, we got 6 phosphoglyceraldehyde in the reduction phase. Then again, one is going to get separated, I said. That means here, 2 is going to get separated. From this 12 phosphoglyceric acid, 10 is going to enter for RUBP regeneration, and 2 phosphoglyceric acids are going to enter for glucose formation. This is the most important thing about this equation. 6 RUBP, 6 carbon dioxide molecule. Then 12 phosphoglyceric acid formation will take place. This will undertake reduction next stage. After reduction, this phosphoglyceric acid is going to become glyceraldehyde 3-phosphate or PGAL. PGAL means phosphoglyceraldehyde. So from this 12, 2 will be utilized for glucose formation, 10 will be utilized for regeneration of RUBP in the third stage. So this reaction is catalyzed by the enzyme RUBP carboxylase. As you know, carboxylation is the first stage of Calvin cycle. Reduction, second stage. The second part of the path of carbon is reduction of phosphoglyceraldehyde. So ATP and ADPH, which are synthesized in the light reaction, will be utilized here. That's why this is called reduction stage. In this reduction stage, the phosphoglyceric acid is converted to phosphoglyceraldehyde, or we can also say glyceraldehyde 3-phosphate. Don't get confused with this terminology of PGAL or glyceraldehyde 3-phosphate. Both are same only. This part includes the following reactions. What is happening in this means first ATP will be added, then NADPH will be added. So first how ATP is added? PGA is phosphoglycerated by phosphoglycerokinase enzyme to yield bis-phosphoglyceric acid. PGA is having only one phosphorus, one phosphate. Now it will have another phosphate added from the ATP. So ATP donates phosphate group and it is becoming bis-phosphoglyceric acid. Now this bis-phosphoglyceric acid is reduced to phosphoglycerodehydrogenase to yield phosphoglyceraldehyde PGAL, or we can also say glyceraldehyde 3-phosphate. So NADPH is going to donate the hydrogen atoms and this is possible by NADPH. So ATP, NADPH are utilized in the reduction phase. Reduction phase, remember, where is the end product of light reaction utilized in biosynthetic phase? Means you must identify reduction phase. In the reduction phase, the phosphoglyceric acid will be converted to phosphoglyceraldehyde. That is by ATP donating the phosphate group for making it bisphosphate, and NADPH donating its hydrogen atoms for making it completely getting transformed to phosphoglyceraldehyde. The other name of this PGAL phosphoglyceraldehyde is G3P. G3P means glyceraldehyde 3-phosphate. So energy rich phosphate group is released. Now the energy rich phosphate group is getting released. Why? Because ATP and NADPH are energy packets. These energy packets are transferred now to our G3P, glyceraldehyde 3-phosphate. So 2 molecules of glyceraldehyde 3-phosphate are used to form starch. Okay. So from this 12 molecules of glyceraldehyde 3-phosphate, 2 molecules will be utilized for starch formation, for glucose formation, and 10 molecules will be utilized for RUBP regeneration. So here once again have a look about the cycle. Carbon fixation is taking place, carbon dioxide combining with RUBP3 molecules, and it is becoming in the presence of Rubisco enzyme, and it is becoming phosphoglyceric acid. 6 molecules of phosphoglyceric acid. 6 molecules of phosphoglyceric acid by addition of ATP and NADPH is getting reduced and finally becoming glyceraldehyde 3-phosphate. Glyceraldehyde. From acid it is becoming aldehyde. Remember, in reduction from acid phosphoglyceric acid it is becoming phosphoglyceraldehyde. Okay. Now the energy from ATP and NADPH is also transferred to this. Now this is having energy and here 6 molecules are there. But from 6 molecules, as you can see, down arrow indicates 1 glyceraldehyde 3-phosphate goes to make glucose. Remaining 5 glyceraldehyde 3-phosphate are recycled. That means they are entering here and getting combined with 3 ATP to become ribulose bisphosphate, biphosphate. It was monophosphate it will become bisphosphate. It was previously glyceraldehyde 3-phosphate was 3-carbon compound. Now all together, by removal of 2-phosphate and then addition of ATP, 3 ATPs, it is becoming 5-carbon compound. It is nothing but the regeneration has taken place and the ribulose bisphosphate is formed again here and ready to combine with another carbon dioxide molecule. I hope you understood and you have checked here. For 3 molecules, 6 ATP are used here and 3 ATPs are used. 6 plus 3, 9 ATPs are used for 3 molecules. 9 ATP and 6 NADPH. If we double it for 6 molecules of carbon dioxide, it is 18 ATP and 12 NADPH are utilized. So remember these calculations you must have in mind. Here when we are writing here 3 carbon dioxide means 6 ATP plus here additional 3 ATP, 9 ATPs are utilized and here 6 NADPH are utilized. When they will ask in any question objective type for 6 molecules of carbon dioxide means you have to double this figure. You have to say 9 plus 9, 18 ATPs, 6 plus 6, 12 NADPH. Okay. So this is the complete setup of carbon fixation. But coming to the reduction is not as simple as you see here. You have to see the equations. 6 molecules of phosphoglyceric acid combining with 6 ATP giving 6 bisphosphoglyceric acid and ADP is separated. And this 6 bisphosphoglyceric acid is combining with 6 NADPH molecule giving 6 glyceraldehyde 3-phosphate and NADP is getting separated with acid here. And next, for 12 molecules also the same thing is mentioned here in this diagram where reduction is taking place. Regeneration, this is most important. Regeneration of carbon dioxide acceptor molecule RUBP is crucial in the cycle. If the cycle is to continue uninterrupted cycles are going to take place, and here again and again RUBP must be regenerated. The regeneration steps require 1 ATP for phosphorylation to form RUBP. So for 6 molecules of RUBP, 6 ATPs will be utilized. Okay. So the glyceraldehyde 3-phosphate, which is a product of reduction, and isomer is also there here, DHAP. DHAP is nothing but dihydroxyacetone phosphate. It is also the G3P only. They are isomers. They will be changing their forms. And the interconversion is catalyzed by triose phosphate isomerase. This enzyme is present, which is going to make the conversion happen. G3P and DHAP will be formed actually, but this DHAP can get converted to back again G3P because of the triose phosphate isomerase enzyme. So 10 molecules of glyceraldehyde 3-phosphate we can say, or 5 molecules of glyceraldehyde 3-phosphate, 5 molecules of DHAP will be there, which are going to undertake the regeneration of RUBP. And this is going to take place one by one in a stepwise manner. Chemical equations are there. How this first 2 molecules of glyceraldehyde 3-phosphate and 2 molecules of DHAP in the presence of aldose enzyme, they are going to get converted to fructose 1,6 bisphosphate. 2 molecules of fructose 1,6 bisphosphate. Now again these 2 molecules of fructose 1,6 bisphosphate in the presence of fructose 1,6 bisphosphatase enzyme is going to become 2 molecules of fructose 6 phosphate. And here the phosphate is removed here. You can see 2 pi means 2 phosphates are removed here. It is 1,6 bisphosphate means having 2 phosphates. By this enzyme, it is getting converted to monophosphate. That means 6 phosphate only and 2 phosphates are removed here. Okay. Next, again this 2 fructose 6 phosphate is combining with another 2 molecules of glyceraldehyde 3-phosphate, another 2 molecules. Okay. And in the presence of trans-ketolase enzyme, it is becoming 2 molecules of xylulose 5-phosphate and 2 molecules of erythrose 4-phosphate. These 2 substances are going to form here. Next, 2 erythrose 4 phosphate. This 2 erythrose 4 phosphate is combining with 2 DHAP, and in the presence of aldolase enzyme, it is becoming 2 pseudoheptulose 1,7 bisphosphate. 1,7 bisphosphate. 2 phosphates are there in this. Again, this 2 pseudoheptulose bisphosphate 1,7 bisphosphate is getting converted to monophosphate in the presence of pseudoheptulose 1,7 bisphosphatase enzyme. It is becoming pseudoheptulose 7 phosphate, and you can see the 2 phosphates are getting separated here. Again, this 2 pseudoheptulose 7 phosphate monophosphate combines with 2 molecules of glyceraldehyde 3 phosphate in the presence of trans ketolase enzyme, getting converted to 2 xylulose 5 phosphate and 2 ribose 5 phosphate. This 2 xylulose 5 phosphate and another 2 xylulose 5 phosphate, both of them, that means 4 xylulose 5 phosphates are going to participate in the next stage, where ribulose 5 phosphate epimerase enzyme is going to work on it, and it is going to get transformed into 4 ribulose 5 phosphate. Xylulose is getting converted to ribulose 5 phosphate in this. Then next step, 2 ribose 5 phosphate, this 2 ribose 5 phosphate in the presence of ribulose 5 phosphate isomerase enzyme is getting converted to 2 ribulose 5 phosphate. Ribose has become to ribulose 5 phosphate. Now you can see here 4 ribulose 5 phosphate is there, 2 ribulose 5 phosphate is there. That means total 6. These two are going to undertake the last chemical reaction, that is 6 ribulose 5 phosphate getting added by 6 ATP is becoming ribulose 5 in the presence of ribulose 5 phosphokinase, becoming 6 molecules of ribulose 1,5 bisphosphate. 1,5 bisphosphate means the monophosphate of ribulose 5 phosphate has now become the bisphosphate, and you can see 6 ADP are released. That means 6 ATP is added here and 6 ADP is formed along with ribulose 1,5 bisphosphate. This is what RUBP regeneration that has taken place. How this RUBP has taken place, you can get back to the previous here. Glyceraldehyde 3 phosphate, DHAP. 2 glyceraldehyde 3 phosphate, 2 DHAP. And here you can see other 2 G3P. 2 plus 2, 4 G3P. 2 DHAP and 2 DHAP, 4 DHAP. Here another 2 molecules of glyceraldehyde 3 phosphate you can see here. Totally these 10 molecules of glyceraldehyde 3 phosphates are going to get transformed into ribulose 1,5 bisphosphate by addition of this 6 ATP molecule. This is what regeneration of RUBP taking place. And here remember, for every carbon dioxide molecule entering the Calvin cycle, 3 ATPs and 2 NADPH are required. Okay, for every Calvin cycle, 3 ATPs, 2 NADPH. For synthesis of one glucose molecule, 6 turns of Calvin cycles are required. So this is what is in, what is out chart, where you can see 6 molecules of carbon dioxide, 18 ATPs, 12 NADPH. These are getting utilized, and when this is happening, glucose formation will be taking place, as well as RUBP regeneration will also taking place. And when this ATP is entering and NADPH is entering, the reduction is taking place. ATP getting converted to ADP, and the same number of NADPH getting again NADP. They are becoming NADP again, and 6 carbon dioxide molecules are utilized, and here one glucose molecule is also formed here, and the regeneration of RUBP also takes place. So this is what about Calvin cycle. I hope you understand this Calvin cycle very well. It is somewhat confusing when many equations are there, but slow and steady if you revise in the textbook also, as well as watch the video again. The diagrammatic representation is very clear for you, where 3 carbon dioxide molecule is combining with RUBP. Have a check about that. Coming to the assignment, explain Calvin cycle. This Calvin cycle, which is having 3 stages: carboxylation, reduction, regeneration. This thing you must learn and be perfect. And the part of this regeneration where chemical equations are given, you must write in a sequence order. 9 reactions will be there. And finally, RUBP will get regenerated by utilizing 10 molecules of glyceraldehyde, 3 phosphate, and 6 ATP. So, with this, we finish this Calvin cycle. Thank you. Thank you. And have a good day. Sir, end of the session. Sir, end of the session. Class is over.